Today, we would like to talk about how gentle cell sorting can lead to very solid and good results in NGS or single-cell genomic analysis. Our talk will be divided into two parts. The second part will be presented by Quy. He will talk about his research on murine mammary epithelial cells and how he utilizes single-cell RNA sequencing technologies as well as cell sorting technologies in order to gain the maximum out of his experiments. In the beginning, I would like to give a short introduction on gentle cell sorting upstream of next-generation sequencing analysis, and of course, I would like to focus on the MACSQuant Tyto by doing so. So let's first have a look on NGS or single-cell genomics application areas in general. As you all know, these both technologies are really important and are utilized in lots of different application areas, starting from basic research in, for example, cell biology, immunology, or developmental biology, but also going into translational or clinical fields such as cell therapy, cancer research, metabolic disorders, or in the moment, very important, also the research on infectious diseases. At Miltenyi Biotec, we are actually focused on providing products which allow for a very gentle and proper sample preparation upstream of NGS and single-cell genomics analysis. I would like to shortly highlight the GentleMax dissociators, which can be utilized if you start with solid tissues such as brain, for example, or tumors, and want to generate a single-cell suspension. But also coming from blood products, we have several interesting products for straining and clearing. Coming to a next step, an optional step, that is of course only important if you're interested in specific target cells, and that is the cell isolation or cell sorting. Cell isolation via our MAX technology, so via magnetic beads, can be utilized. Here we have an example for tumor-infiltrating leukocytes, which have been enriched based on their expression of CD45. If you're interested in multi-parameter analysis or sorting of the cells, flow cytometry is a very good way to go. And here we offer the MACSQuant Tyto cell sorter in order to sort your cells based on multiple parameters on a very gentle way, so you get out very viable cells in very high purities, as you can see in that example given for the natural killer cells. Coming from those technologies then to your downstream analysis, you can proceed with your NGS or single-cell genomics analysis. And today we of course would like to focus on the MACSQuant Tyto, so the cell sorting step prior to NGS or single-cell genomics. So why is it in general important to sort your cells prior to these analysis? Of course, the very obvious reason is you're interested in enriching a specific target cell population and thereby to improve your reads. This can be done based on expression of specific markers on the cells, but also based on their size or granularity. Another important factor why cell sorting should be applied upstream of single-cell genomics analysis is the exclusion of dead cells and the removal of debris. Both can actually contribute to a high background by extraneous DNA or RNA contaminations, for example, and thereby lead to high costs. So you can actually decrease your costs by sorting your cells upstream of your very expensive single-cell genomic analysis. But cell sorting also can come with some associated problems. First, lots of the cell sorters which are currently available in the market have a very harsh sorting procedure, the so-called droplet cell sorters. These can, dependent on your cells of interest, again lead to dead cells, and those dead cells might end up in your precious sample. And if you're only analyzing, let's say, 10,000 cells in the end, if you have 60% viability only, you have already 40% of dead cells in, and this also leads to very high costs in the end. And of course, DNA and RNA contaminations can also here lead to high backgrounds or false results. A second problem is that due to the stressful sorting, gene expression artifacts can be generated, and that might also lead to false results, specifically if you're interested in markers around stress, activation, or metabolics, for example. Another important factor to consider is timely delays. A very difficult and slow processing might also then again lead to gene expression alterations in your target cells and thus again to false results. So it is all about circumventing and these problems, and we think we have a very nice solution with a MACSQuant Tyto cell sorter. So I would like to introduce to you the instrument itself. The Tyto, as you can see here, is a benchtop cell sorter with a very small footprint. It comes with two different screens, and a very important factor is actually that you can control the sorting temperature between 4 and 25 degrees of Celsius. I said before, we can sort our cells based on multiple parameters, up to 10 in the MACSQuant Tyto. The instrument comes with 3 different lasers, and you can sort based on up to 8 fluorescence channels and 2 scatter channels, which provides full experimental flexibility and a possibility of multi-parameter sorting. Unfortunately, due to time reasons, I cannot go into too much details concerning the specifications of the instrument, but if you're interested, please visit our website. There you can find our image brochure or the user manual, which contains all the necessary information. Or you're always welcome to contact us. Here you can see the MACSQuant Tyto cartridge, where all the sorting happens. So the cells won't leave this cartridge during the sorting process. We divide 3 different chambers in this cartridge. On the left-hand side, we have the input chamber, where your cell suspension of interest is filled in. In the middle, we have a positive collection chamber, where the positively collected cells will end up. And on the right-hand side, we have the negative collection chamber, where all the rest will flow through. It is important to mention that we don't have any waste, so you can always regain your cells from the negative collection chamber and run them again through another cartridge if you want to sort another population out of your sample. A mixing propeller in the input chamber prevents the cells from settling down. Where all the analysis and the sorting actually takes place is the microchip at the bottom of the cartridge. And on the right-hand side of the slide, you can see on the bottom view and in a zoom the microchip itself. I would like to show you this comparison here to make you aware like how small the microchip really is. And if we zoom in, we see that there's actually some channels inside this microchip. So if the cells come from the input chamber onto the microchip, they enter this on the lower left-hand side. They run through the sorting channel where they also pass the lasers. They're analyzed, and in a case of a positive event, a magnetic pulse opens a microvalve, which then simply redirects the cell into the upper channel, so the cell ends up in the positive collection chamber. We can have a look on that specific valve. You can see it here. So in this position, the valve actually is closed. All the sample flows through into the negative collection chamber. Now, if a magnetic pulse is induced, the valve will open and very gently redirect the cell so it ends up in the positive collection chamber. To get a better understanding like how this process really is working, I would like to show you a short video on that so you can really understand like where the cells are redirected and how they flow through the microchip. So now that you have seen this animation and also the video of the very, very fast microvalve, an opening and closing cycle only takes around 30 microseconds. So the microvalve can move up to 30,000 times per second. It became quite obvious that this is a very, very different cell sorting technology if you compare it to the classical FACS, the fluorescence activated cell sorting droplet sorters. You can see a picture describing the technology here on the right-hand side on the slide. And this technology comes with some problems. First of all, droplet sorters are always open systems. So the aerosols and droplets which are produced are in an open space, which of course is a problem if you are, for example, interested in going into clinical applications, but which can also be a problem, for example, in terms of contamination. Contaminations and sample-to-sample carryover can also derive from fluidics and tubings which are shared between the samples and which are inside the instrument. So the sample has to flow through the instrument in order to be sorted. Another problem coming with this kind of technology is the very harsh sorting process. So the sorting takes place at very high pressures in these systems, which depending on the nozzle size can go up to 70 psi. And these are followed by a very strong decompression once the droplets, including the cells, leave the nozzles. Directly after that, a charge is applied to the cells in order to redirect the droplets in an electric field. And this is also another stressful event for the cells. And in addition, strong shear forces act on the cells during the sorting process. And all of that can, in the end, lead to very high cellular stress levels, low viabilities in dead cells, also a low final cell yield, belt functionalities in the cells, but also to degeneration of artifacts. Now, if you compare with a cell sorting technology in the MACSQuant Tyto, which is microvalve mediated, we have a very gentle cell sorting technology here, acting on a very low pressure of only 1.5 to 2 psi. There's no decompression, no charge applied to the cells, and only mild shear forces acting on the cells. And this results in highly viable and functional cells after the sorting process. But also the system is very safe, and that is due to its closed and sterile cartridge environment. There's no carryover from sample to sample, as the cells won't leave the cartridge during the sorting process. There are no fluidics inside the system. And so no contaminations can come from the instrument into the cartridge. But also for the operator, it's a very safe cell sorting system. On the one hand, as I said, it's closed, but it's also not producing actively any aerosols. And this might come in very handy if you're sorting infectious materials such as virus-infected cells, for example. So all in all, we have a very gentle way to sort cells using the MACSQuant Tyto. And on the next slide, I would like to show you a setup we actually built in order to film the cells which are actually arriving in the positive collection chamber. And therefore, we put an endoscope inside the positive collection chamber. And you can see in a video, which I will show shortly, see how the cells flow into the positive collection chamber in a very, very gentle way. But I'm also showing this video to make another point clear, and that is that the cells are actually strongly concentrated during the sorting process, because the valve only opens in that moment where the specific target cell is at the valve and then directly closes again, meaning the main portion of the buffer will flow into the negative collection chamber. And depending on your downstream process, for example, how you perform your library prep, if you, for example, work with 10x chromium, this might be interesting because it could potentially save you a step of centrifugation. I would also like to shortly comment on the workflow we see in the we apply in the MACSQuant Tyto. Of course, it is a flow cytometry-based cell sorter with up to 10 parameters. So this means, of course, we have you have to think about your gating strategy and the antibody panel you want to use. But once you've set up your experiment, you can save the workspace, and then the workflow is very, very easy and straightforward. And that is simply, you have to load the sample into the cartridge. You scan the cartridge, which has a barcode, and so all the settings are already set directly in the software. You load the cartridge into the temperature-controlled loading chamber, and then you just press play and can walk away. It's an operator-free sorting process. You don't have to stay in front of the instrument and check, for example, for drop delay or something like that, because this is simply not needed in the Tyto sorting system. Once the cells have been sorted, you can come back, collect your sorted cells, and proceed with your experiment. And in a direct comparison, this can lead to hands-on time, which is up to 9 times less compared to a conventional droplet sorter. And with that, I come to the last part of my introduction, and that is I would like to give you some application examples before Quy talks about his research, just to discuss again how gentle the cell sorting process on the MACSQuant Tyto really is. And with this first example, I would like to discuss the sorting of neutrophils. And everyone who already worked with neutrophil granulocytes knows that these cells are really, really sensitive, really fragile. They can be easily activated. They will also die very easily, so you have to be very careful with handling them. Now here you can see the input and the sort. We were able to sort the neutrophils to a high purity of 98%. But very important, the cells also stayed viable after the sort, 98% viability, which is already great, but doesn't necessarily tell us if the cells are still functional or might have been activated. So we checked the surface expression levels of some proteins, which are either up or down regulated during neutrophil activation. And as you can see, the samples in the MACSQuant Tyto, which is the orange bar, are comparable to the one of the negative control, which is the white bar. But also functionality of these cells was still fully intact. So we checked, for example, for phagocytosis or the migration towards a chemokine gradient, also called chemotaxis, in this case, a FMLP gradient, both very complex processes. A lot of signal transduction is involved and mechanical interactions of proteins. And as you can see, both are still working very well. And this proves that also these cells have been sorted using the MACSQuant Tyto as a flow cytometry-based cell sorter. They were still fully functional and not activated after the sorting process. A second example I would like to discuss is about sorting of iPSC-derived dopaminergic progenitors. This data was kindly provided by Daisuke Doi from the Saira Institute in Kyoto, Japan, and is actually deriving from a clinical study they are currently running in order to treat patients suffering from Parkinson's disease. And they were first working with a FACS droplet sorter, but encountered some difficulties in translating into the clinics. With the Tyto, this is much more easier because of the closed and sterile sorting environment. We also offer our consumables in a GMP-compliant format, so that was already great for them. But they also wanted to check the gentle aspect of the cell sorting because they had some problems with the expansion and the yield of their cells after the sorting process. So what you can see here is an analysis 13 days after these iPSC-derived dopaminergic progenitors had been sorted, and you can see these cells are forming spheres. And the spheres in the MACSQuant Tyto sorted sample are much bigger. And this is highly significant and comes hand in hand with a much higher cell yield of 160% compared to the other sample. And actually is due to very functional, viable cells So we have a high cell proliferation and expansion possible in the MACSQuant Tyto. But it's not only about cell growth, it's of course also about the engraftment of the cells, and you can also test this in vitro by checking the area of neurite extensions, which are actually an indicative of graft survival capacity of these cells. And again, you can tell by eye that this area is bigger in the MACSQuant Tyto sorted samples if you compare to the FACS droplet sorter. And of course, this goes hand in hand with a higher graft survival capacity, which is of course key for a successful cellular therapy. And that is also why the group now uses the Tyto in its clinical trial, the first in-human iPSC-based clinical trial to treat patients suffering from Parkinson's disease. And if you're interested in more details on that, we can also provide a poster which had been presented by Daisuke Doi at the ISSCR in 2019. And this brings me to the last slide of the introduction, and it is just that I would like to mention that using the MACSQuant Tyto, you cannot only sort normal mammalian cells, you also can sort other stuff. We frequently sort, for example, bacteria or yeast, but you can also sort nuclei, which is of course very interesting for applications in the NGS and single-cell genomics context. And here I wanted to show you an example. You can see the input and the respective sort fraction. We were able to enrich the nuclei based on their staining on DAPI and marker I can not actually talk about because this is data we also got from a customer and it's confidential, but you can see in general we were able to enrich these nuclei to high purities. And with that being said, I think it's time to hand over to Quy so we can learn more about his research and how he utilizes gentle cell sorting technologies in his workflow. And yeah, with that, Quy, I'd like to hand over to you, and I'm looking forward to listen to your presentation. Thank you very much.
Thank you, Felix. Okay, so welcome. I'm Quy Nguyen. I'm in Dr. Kai Ketzenbrock lab at the University of California, Irvine, and we work on profiling the normal breast development and how that leads to, or how the development of breast cancer is initiated, mainly in human. We've also done a lot of work in mouse, and today I'll talk a little bit about our efforts in that. So a little bit of background on why we do what we do. So with the Mammary, so today I'm going to talk about the mouse system, and with the Mammary gland development in mice, at the very top panel, this is taken from a review, you can see that it's mainly a fat pad that epithelial cells grow into, almost like a tree structure. And with that, you can see that the main cells that make up, or the cells of interest to us, are the epithelial cells, and that is because those epithelial cells are believed to be the cells of interest, the initiating cells for breast cancer. And as you can see, the bottom panel, there's a lot of supporting cells within the mammary gland. There are adipocytes, there are other stromal cells such as fibroblasts, there are immune cell, macrophage, T cell, B cell, there are mast cells, red blood cell, all different kind of cell types in there. And if we really want to study just epithelial cells, what we have to do is enrich for these cells somehow and continue to profile them. So in a previous recently published paper, it's shown that there are three major cell types within the epithelial system, or the epithelial cell. And you can see there's one basal population and two luminal cell types. And within that, there are multiple cell states. So what we want to do is be able to reconstruct this within the mouse system. And to give you guys a little bit of background on our approach before and what we're currently testing is what we would do is extract out the mammary gland from these mice, mechanically dissociate them down to almost a single cell, pretty much a single cell suspension. And what we would do is FACS isolate these cells for epithelial cells before and run them on the 10x. What we were interested in seeing is there another approach to reduce the stress that FACS induce on these cells? So what we did in this test was take the same cell suspension, split it into two pools, one where we ran onto the FACS machine and the other we ran on the Tyto machine for sorting. And then we ran both sample onto the 10x right afterward just to see the profile. So why would we want to take the single cell approach for this? As you can see on the left, when all of the previous work that has been done using bulk approach, you can see where it's just, you're just taking a sample of an average sample, so you can't really tell heterogeneity at the cell state or type level, where when we do it on the single cell level, as you can see on the right there, you can tell which cells are high for which gene, which cells are low, and be able to distinguish on the single cell level which cells are of interest and which cell are not, instead of just pulling them all together and taking an average of everything. So the sequencing approach that we take is the 10x genomics approach with using their RNA 3' sequencing platform. And what that does is it takes in a single cell suspension, it generates droplet, it partitions these cells with barcode into droplet for us to be able to generate a cDNA library that's unique, that's barcode unique for each cell, for us to later be able to bioinformatically distinguish which transcript came from which cells. And as you can see with the 10x approach, where it's partitioning cells into these droplets, it's very important for you to start off with a very clean sample that are single cell. If they're not single cell, you're going to get multiple. If they're sticking together, you're going to get a multiple droplet, and that will throw off your data. Or if there's a lot of ambient RNA that's put into this platform, that would also be captured, and you can't. It's very hard to tell what was ambient RNA and what came from the cell. So to show you guys the importance, so these are two samples that were acquired. One on the left was not sorted, and one on the right was sorted. And you can see this is an output from Cell Ranger where it shows cell calling. And the sample that was not sorted had very high contaminating ambient RNA. And you can see it takes about, only about 50% of the sequence read were actually used or called in within the cell, while the sorted cells had about 97, 98% of the reads in cell. So with an unsorted sample, you're wasting about half of your reads, your sequencing read, and at the same time, it makes it very hard for the program to tell which are cells and which are just empty droplet containing ambient RNA. And in addition to that, all of your droplet that contains actual cell also contain ambient RNA to sort of cloud your real readout. So this has been shown before, but I'll go over it again with our setup. What we did was we did this in triplicate. And with the FACS sorter, we used two different FACS machines just to see if it's reproducible. And the PSI that we use 100 micron nozzle, and that produces or puts the cell under around 20 to 25 PSI of pressure. And while the FACS sorted cell, when we ran it through the cartridge, was around 1.5 PSI. So there's a big difference. And in addition to the FACS, the cells being compressed with about 20 PSI is the rapid decompression as it comes out of the nozzle to be collected into the collection tube. So this is the overall data with all 6 samples, so 3 replicates, so 3 for FACS and 3 for Tyto. We collected around almost 48,000 mammary epithelial cells from the mice, and you can see what we reproduce, as shown before in human, are the three major cell types. So one basal and two luminal population. And you can see on the right there, the top 10 marker heat map for each of these cell types, or you can see that they, between the FACS-sorted cell and the Tyto-sorted cell, have very similar gene capture. So we're not losing too many important cells for either of these methods. So to go into a little bit more finer detail analysis, what I did was I subsetted on individual cell type, so basal, luminal 1, luminal 2, and did a comparison for their marker gene. And as you can see on the right there, there isn't too big of a difference. There isn't actually any difference between the FACS and Tyto-sorted cell for the marker gene. So we're able to pick up traditionally what we would in FACS as we do in Tyto. And on the left there is a t-SNE plot colored by the replicate. So you can see that the three replicates for our experiment overlaps very well. And this is clustered without any batch correction. So we can see that we're not introducing anything major with Tyto-sorted cell or FACS-sorted cell. But the difference you can see really comes down to when you take a look at the content of these cells, or sort of what they're expressing. So when we take a look at numbers of gene capture, UMI, percent mito, you can see that's where it stands out a little bit more. With the FACS-sorted cells, we capture definitely more genes. So it's around 1,400 cells for Tyto-sorted cells, or 1,400 genes for Tyto-sorted cell, and around 1,200 genes for FACS-sorted cells. And when you move over to the UMI, so the unique molecular identifier, the molecule, we do capture more with Tyto cells, so around 3,200 UMIs for Tyto and around 2,400 for FACS. And for percent mito, it is a little bit higher in Tyto, and we believe that could be due to just when we run the 10x sequencing, we're capturing more gene, we're capturing more transcript, hence why we're capturing a little bit more of the percent mito, or more of those mitochondrial genes. So there was a paper that was published where they take a look at sort of a dissociation-induced signature in their sort of protocol, and sort of see is there a signature and is that reproducible. So what I did was I took a look at that signature and scored it on our data set just to see, is it higher in the Tyto, is it higher in the FACS, or is it similar? And when we take a look at those genes, we can see that the FACS-sorted cell has slightly higher expression of these genes compared to Tyto. And that was one set of genes. So what I wanted to do was went online, acquired sets of genes that are involved in multiple pathways, such as apoptosis, necrosis, autophagy, cell death, everything else that you see here. And what you can see is that when I scored these these sets of genes for cells that were sorted on the Tyto machine or cells that were sorted on the FACS machine, you can see that FACS-sorted cell have slightly higher expression of these, or scored slightly higher for these pathway, and for a majority of these pathway. So that was very interesting. What we wanted, that was just sort of on the gene expression level. What we wanted to see was, does that carry over? Is there something functionally different with these cells? So taking those same exact cells, actually, that were dissociated, split, stored on the FACS machine, Tyto machine, we took those same exact cells after running it on the 10x, or same pool of cell after running on the 10x. We split off some and we seeded these cells in matrigel to run a Mammosphere formation assay to see, do we get more sphere? Do we get larger sphere? Is there a growth difference from this long term? And you can see here on the left is a quick illustration, an actual image from our Mammosphere after 7 days. You can see that Tyto-sorted sphere on the image on the left there there's more sphere compared to the FACS-sorted cells. on the right. And the bar graph on the right there is a quantification for our experiment. And overall, you can see that long-term experiment, you can see that Tyto cells have much more sphere growth compared to the FACS-sorted cells. So quick summary for everyone. So with our data set, our experiment, we detected 3 major cell types as previously seen in human. And what's very nice is that we see a similar cell type between FACS and Tyto. So with FACS, we're not losing any important cell type. We're not gaining anything too much with either one. But what's interesting, especially for next generation or the single cell experiment, is that with Tyto-sorted isolated cells, we actually see higher gene and UMI capture for these cells, while with the FACS isolated cells, they're introduced with a lot more stress throughout the entire process. So we do see a slight induction of these stress response genes. And functionally, what's very interesting is we see that Tyto-sorted cells form more Mammosphere, and that's very important for long-term experiment or if these cells are of interest and you need them to grow up. So thank you.